Good afternoon to everyone. Welcome back to the YouTube channel ePathshala. Please subscribe ePathshala YouTube channel and share. Okay, this is Ramesh, Faculty of Biology. Okay, dear students, in previous session we discussed about cell, the basic unit of life lesson. In that lesson we discussed multicellular organisms and unicellular organisms. At the same time, we observed the mastic cells under the microscope. Okay, today I am going to explain about observing onion peel cells. Okay, today I am going to continue that lesson. Listen carefully. Dear students, actually in previous session we discussed unicellular organisms, multicellular organisms. Unicellular organisms means single-celled organisms. Examples also we learned. And multicellular organisms are made up of more than one type of cells. The examples are fungi, plants and animals. So at the same time, now I would like to introduce the prokaryotic cells and eukaryotic cells. So based on the nuclear membrane, the cells are two types. They are prokaryotic cells and eukaryotic cells. So what are the prokaryotic cells and what are the eukaryotic cells? Cells that have membrane bound nuclei are called eukaryotics. Again I am repeating. Cells that have membrane bound nucleus. It means a round bound nuclear membrane. Okay. So cells that have membrane bound nuclei are called eukaryotic cells. Means true cells. So if the cell contains true nucleus, they are eukaryotic cells. If the cell contains true nucleus, that is eukaryotic cells. So in that eukaryotic cells, we can see the nuclear membrane very clearly. So the nuclear membrane is visible in that eukaryotic cells. So based on this nuclear membrane, these cells are prokaryotic cells and eukaryotic cells. So have you understood about eukaryotic cells? Again I am repeating eukaryotic cells. So cells that have membrane bound nuclei are called eukaryotic cells. The examples of eukaryotic cells. What are the examples of eukaryotic cells? So the examples of eukaryotic cells are protists, plants, animals and fungi. Protists, plants and animals and fungi. Protists, plants, animals, fungi. Okay, protists, plants, animals, fungi are the eukaryotic cells. Eukaryotic cells. So what are the prokaryotic cells? Again I will explain. So what are the prokaryotic cells? So cells that lack of nuclear membrane. Cells that lack of nuclear membrane are called prokaryotics. So these cells didn't have nuclear membrane. A proper nuclear membrane is absent in these prokaryotic cells. Again I am repeating. Cells that lack of nuclear membrane is called prokaryotics. Examples are bacteria, blue green algae, mycoplasma, ppl. So before going to explain these eukaryotics and prokaryotic cells, I wanted to introduce about Cells, actually dear students, the cells are two types, plant cells and animal cells. In previous class we learned about there are two scientists. So Sladen and Swan, who? Sladen and Swan, they formulated the cell theory. So the Sladen explained about plant cells. The plant cells are he found the plant cells in the plants. Theodore Swan found animal cells in animals. So like this way, the plant cells, animal cells, there are two types of cells, plant cells and animal cells. So actually these cells are, we can't see with our naked eye. So how can we see these? Yes, we can observe these cells are under the microscopes. So plant cells and animal cells. So one of these cells, plant cell, I drawn the diagram. The plant cell diagram I drawn on the board. So once observe it carefully, and what may be the parts are present in these cells, and observe clearly and keenly, because of very most important, the differences between plant cell and animal cell. So number of differences are takes place. So in those the cell wall, the plant cells contains cell wall, the animal cells doesn't contain cell wall. We can see a big sized vacuole in plant cells. In animal cells, if the animal cells contains vacuoles, very small in size. Some animal cells didn't have vacuoles. So plant cells contains big sized vacuole. So in this plant cell, the nucleus is present in the corner of the cell. In the corner of the cell, the nucleus is present. But in animal cell, the nucleus is present in animal cell, center of the cell. So there are the differences. The chloroplasts are present in plant cells, absent in animal cells. Okay, these just some differences. The cell wall is present in plant cell. Cell wall is absent in animal cell. The nucleus is present in corner of the cell in plant cells. So the nucleus is present in animal cells, center of the cell. The chloroplasts are present in plant cells, absent in animal cells. Once look at the diagram and observe keenly. Okay, so this structure is a circular structure there, no? They are plasmodesmatas. The plasmodesmatas are present in these areas. And next, cell wall. Next, plasma membrane. These are the lysosomes. And next, free ribosomes. They are the Golgi complex or Golgi apparatus. These small circular structures are Golgi vesicles, and these are the smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum. SER. And then this is the rough endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes. So a round bound nucleus. So this is nucleus. In the center of the nucleus, a small round like structure there, that is nucleolus. So some thread-like structures, a fine strands-like structures are present in middle of the cell. They are the genetic material, means DNA. And then the nucleoplasm is present in this nucleus. This is the nuclear envelope. Some pores are there, nuclear pores. And chloroplast, this is the chloroplast. And next, peroxisome. What is peroxisome? And next here, this is the intermediate filament and microtubules, microfilaments. So these structures are belongs to cytoskeleton. So this is the structure of plant cell. So more than details I will explain in the next class. Okay, I will explain about plant cell and animal cell. I will tell you the differences between plant cells and animal cells. Okay, have you observed clearly? Yes, is it okay? Yes. Now I am swiping the board. Just coming to the point, eukaryotic cells and prokaryotic cells. So already I explained you what are the examples of eukaryotic cells: protists, plants, and animals, fungi. So protists, I think you don't know protists. So Protista means protozoan organisms. So protist is one of the kingdom. Actually, there are different kingdoms are there. So 5 kingdoms, 2 kingdoms, 3 kingdoms, 6 kingdoms. Different scientists are discovered. Different scientists are discovered in the different years the kingdoms. So actually, we well known about Whittaker kingdom. So the Whittaker kingdom is the 5 kingdoms. So I will explain it clearly. Okay students, this is history of classification. So here Linnaeus, one of the scientists, Haeckel, Charton, Copeland, Whittaker, Woos et al., Cavalier Smith. So they are the different biologists. So in the history of science, in India, classification had been the basis of the studies in medicines, and dates back in 1st and 2nd century. So AD. And next Charaka, do you know Charaka? Charaka and Sushruta had classified the plants on the basis of their medicinal importance. So thereafter Parasar in his book Ayurveda, the science of life trees, documented the classification system for several land plants. So for the first time, this classification mainly deals with the structure of the flowers. So Charaka and Sushruta classified these structures based on the structure of flowers. So let us study how biologists in 16th century have been trying to classify diverse organisms and so forth. So this is the classification done by different biologists up to now till date. The following table shows how the different biologists have gone about forming the first category of a classification. So different scientists have tried to classify plants and animals. Some scientists are classified plants based on their structure and functions and their medicinal importance, and animals also based on the body size, based on the their functions, their structures, the animals are divided. So like this way the biologists are classified these classifications. Some scientists classified 2 kingdoms. So Linnaeus classified 2 kingdoms, and Haeckel classified 3 kingdoms. Chatham classified 2 empires, and Copeland classified 4 kingdoms, and Whittaker classified 5 kingdoms, and Woos et al. Classified 3 kingdoms, and Cavalier-Smith classified 6 kingdoms in the year 1998. So Linnaeus 1735, Haeckel 1866, Chatham 1925, and Copeland 1938, Whittaker 1969, and Woos et al. 1990. So in these years they classified. Although biologists in the 16th and 17th centuries did not recognize that the similarities and differences among organisms were consequences of evolutionary mechanisms, they still served a means of organize biological diversity. In 1758, one of the scientists, Carolus von Linnaeus, proposed a system that had dominated classification for centuries. Linnaeus gave each organism 2 names. So this is another topic, but I will give you a small introduction. And denoting genus and species, such as Homo sapiens, the former representing genus while the latter representing the species. He then grouped genera into families, families into orders, orders into classes, classes into phyla, and phyla into kingdoms. So Linnaeus identified 2 kingdoms, Animalia and Plantae. Animalia belongs to animals, Plantae belongs to plants. So all the trees like species, genus, family, order, class, phylum, etc. were defined by Linnaeus on the basis of the similarities and differences. So for the based on these structures, the Linnaeus classified. The Linnaeus gave 2 names for the organism. The Linnaeus gave two names for the plants and animals. Okay, have you understood? So the first major breakthrough from the Linnaeus model came from the Thomas Whittaker in 1969. Whittaker proposed a 5 kingdom system. In this, once look at this. Linnaeus, this is the Linnaeus kingdoms. Linnaeus kingdoms, 2 kingdoms, Vegetalia and Animalia. So Haeckel kingdoms, 3 kingdoms, Protista and Plantae, animals. And Chatham kingdoms, Chatham empires, 2 empires. Prokaryota, Eukaryotic. So already discussed with you prokaryotes and eukaryotics. So prokaryotics, eukaryotics are found by Chatham. So this is classified by Chatham. And next, Copeland. Copeland classified 4 kingdoms: Monera, Protista, Plantae, Animalia. And Whittaker classified 5 kingdoms: Monera, Protista, Plantae, Animalia. And Woos et al. Classified 3 kingdoms: Bacteria, Archaea, Eukarya. And Cavalier-Smith classified 6 kingdoms: Bacteria, Protozoa, Protista, Plantae, Fungi, and Animalia. So these are the classification. This is the classification. This classification is done by different biologists. Okay, have you understood? So in prokaryotics, eukaryotics, the eukaryotics example, one of the example is Protista. I told you that is. So once look at that is prokaryotics, and so once come back. So about prokaryotics and eukaryotics, so there are various hypotheses on the origin of prokaryotic cells and eukaryotic cells, because all cells are similar in nature. So it is generally thought that all cells came from a common ancestor cell termed the last universal common ancestor. Last universal common ancestor, Leuka. That is Leuca. L-U-C-A. Last universal common ancestor. So these Leuca eventually evolved into three different cell types. So each representing domains. So the three domains are the archaea, the bacteria, and the eukarya. Okay, archaea and bacteria are prokaryotic cells as they do not have a membrane bound nucleus. The nuclear material is depressed in cytoplasm. The cell walls of bacteria, unlike the archaea, contain a fat-like chemical peptidoglycan. The bacterial membrane is made up of with peptidoglycan. So eukarya have eukaryotic cells with a membrane-bound nuclei. So if the nucleus contains bound membrane, that is eukaryotics. If it is absent, prokaryotic cells. Okay, once wait. What is that? Yes. These classifications. In these classifications, different kingdoms are there and empires are there. So one of the kingdom name. So yes. First, I wanted to explain about this is Whittaker classification. Whittaker classification is very most important. That's why I wanted to explain some characteristics of these five kingdoms. Whittaker kingdom. So the Whittaker classification: Monera, Protista, Plantae, and Animalia. So these are the five kingdoms classified by Whittaker. Monera. First one, what is Monera? So the Monera kingdom characteristics. So some common characteristics. These Monera organism cells have no membrane and bound nucleus. So they reproduce by splitting into two organisms. So they absorb nutrients from outside their borders. Another animal bodies. So they move with the help of locomotory organs. Monera organisms have some locomotory organs like cilia, flagellus. These locomotory organs. So one of the best example of Monera is bacteria. Okay. And next, what is that? Protista. What is Protista? So what are the common characteristics of Protista? Listen. Say characteristics of Protista. Mostly they are one cell, single celled organisms. Protista organisms are single celled organisms, means unicellular organisms. So these organisms have a membrane around the nucleus. Protista organisms have a membrane around the nucleus. So some get nutrients and energy by eating other organisms. So these Protista organisms eats other organisms. So some get energy from the sun and nutrients from the water around them. So these live either solitary or colonies. So some of the cell organelles are present inside of the cell. So most of the organisms reproduce by splitting into two cells. So the examples of Protista: Paramecium, amoeba, algae, kelps, diagrams. Kelps, do you know kelps? So sea kelps. These kelps are there in oceans and seas. So seaweeds, kelps are also called as seaweeds. Very useful, these kelps. And next, students, fungi. What is fungi? So what are the common characteristics of fungi? So mostly they are many cells, I mean multicellular organisms. Some of the fungi are unicellular organisms also there in fungi kingdom. So eukaryotes with well-defined prominent head. So they get nutrients and energy by absorption, digesting the surface of where they are living through their thread-like parts of their body. So most of these fungi organisms reproduce by spores. Examples: yeast, mushroom, and bread molds, and lichens or lichens. So yeast, bread molds, mushrooms, and lichens are the examples of fungi. Our next, plantae. What is plantae? What are the common characteristics of plantae? Plants are diverse in nature. You well know about the basis of classification, classifying them, the way they acquire the food, the type of reproductive structure they have, and the way they reproduce. They are multicellular organisms. Plants are the multicellular organisms. They prepare their own food by the process of photosynthesis. So this is about plantae. Okay, clear? Next, about plants, the next level of classification based on the whether the differentiated plant body has special tissues for the transport of water and other substances within it. Further classification looks at the ability to bear seeds. Okay, this is the first level of classification. This is the about plants. Okay, plants like mosses, cycas, and ferns, which do not produce flowers and have sporangium or reproductive structures, are called non-flowering plants or cryptogams. So whose produce flowers are phanerogams. So do you know some flowering plants and non-flowering plants are there? Flower and non-flowering. So the examples are pine, neem, mango, etc. So among flowering plants in which seeds are enclosed within fruits are called angiosperms. Example: mango. Okay, these are the different characters, and those in which the seeds are not enclosed by the exposed to outside, they are gymnosperms. Okay, some type of examples: pine trees. So these are the some common characteristics of the plantae kingdom. And next, animalia. The animalia, the classification, the different animals classifications takes place in these animals. So the classification of animals, there are organisms which are eukaryotic, multicellular, and heterotrophic. Their cells do not have cell walls. Most animals are mobile, means they can move from place to another place. So they are further classified based on differentiation in the extent and type of the body design form. So there are some major groups of animalia: porifera, coeluterata, Nidaria, Platyhelminthes, Nematoda, Annelida. So these number of groups of animals are present in animalia. So these are the structures of animalia. So next, dear. So this is the about different kingdoms. So 5 kingdoms. Up to now we discussed about 5 kingdoms of Whittaker classification. So in these 5 kingdoms, all the maximum, moreover, all the common characteristics we discussed about in this. So next, coming to the point, in previous session we discussed about the mastic cells under the microscope. Now I would like to explain about observing onion peel cells. Collect one onion and remove the peel of the onion, and next take it to position. So peel an onion and cut out a small fleshy like portion of the onion bulb, and break this into 2 small parts and try to separate them. Wait. Okay, onion. So select an onion and remove the peel of the onion and then cut it into 2 equal parts. So in middle of the onion you have to cut down the small portion of the onion. So when you collected this small portion of onion, so make it into 2 equal parts. While you are breaking, in middle of these 2 equal parts of the onion, a translucent membrane will appear. So in onion, so you have to try at your home. Here onion is not available. So take it out this onion peel and place it on the slide. So before placing the onion peel on the slide, so make it sure unfolded. So when you place it on the slide, put a drop of water, put a drop of glycerin and saffron in, and observe under the microscope. Okay, if you observed under the microscope, already you know about how to use microscope. I explained in the previous class. How to make the slide also I explained in the previous class. So just I am explaining about this onion, how onion peel cells observe under the microscope. When you place it on the slide, so if you observe this slide under the microscope, some rectangular shaped like structures will appear under the microscope. How these cells are looking like that, I will draw on the board. Wait. Okay, look at the board. So if you observe these cells, already explained you. So whether the plant cells and animal cells, while you are observing, so observe it carefully and keenly, because of some differences are takes place between plant cells and animal cells. So plant cells contains the nucleus is present. The nucleus is present in plant cells, corner of the cell. So in animal cells, the nucleus is present in center of the cell. So here these nucleus are present in corner of the cell. The cells are looking like a rectangular shape. Have you understood? So this is the structure of onion peel cells. So I observed these onion peels under the microscope. When I observed, so the structures are appear like this. While you are observing under the microscope, so draw the figure of what you have observed under the microscope. So this is the onion peel cells, observing onion peel cells. Observing onion peel cells. Okay, and next observing human cheek cells. Okay, now observing human cheek cells. Same process. So now prepare a slide in your own way. So how it is? First of all, wash your mouth cleanly and take a clean wooden spoon or plastic spoon. Don't use stainless steel spoon. And next, take this wooden spoon and scrape it inside of the mouth. Scrape it inner surface of your cheek, like this. Scrape it inner surface of your cheek. And while you are doing this, keep 2 things in your mind. Firstly, wash the spoon thoroughly, and next wash the spoon thoroughly before using it. And next, secondly, do not scrape too hard. Or else you could hurt yourself. And next, now take the scraping that you have collected and place on this slide, and put a drop of water and cover with cover slip. Cover slip is a thin transparent glass, round, circle-like structure. So cover with cover slip and observe the under the microscope. So draw the diagram of what you have observed under the microscope. I observed under the microscope. I draw the diagram. So that picture, that slide is visible to me like this. So human cheek cells means we are the humans. That means we are belongs to animal groups. So this cells are animal cell. Here is this is the animal cell. So in animal cells, where the nucleus is present, the nucleus is present in animal cells, center of the cell. That is center of the cell. So this type of observation is required. So the differences between plant cells and animal cells are: animal cells doesn't contain cell wall, plant cells contains cell wall. So the vacuoles are big sized vacuoles are present in plant cells, absent in animal cells. The chloroplast is present in plant cells, absent in animal cells. So centrosomes, centromeres are present in animal cells, absent in plant cells. So like this way, so the differences are takes place, and more than details I will explain you in the next session. So I think you understood. Okay, if you have any doubts, ask me.